This figure is what I would call the big picture. Um, Sometimes we get so wrapped up in the minor details of how a neuron works and look at all the different ion channels and how that membrane potential fluctuates during an action potential. We forget the whole big picture. So this is the big picture. Action potential is a way to transfer information from one place in the body to another place in the body and then allow that information to be processed. We can make a decision based on that information and then cause a response. So this is a very simple example that requires many neurons that need to get stimulated and propagate action potentials via the nervous system. So here we have a mechanical stimulus occurring at the fingertips. That mechanical stimulus is pressure due to the pen being held. That pen, that pressure, that mechanical stimulus is opening mechanically gated channels on the dendrite of this sensory neuron. That blue arrow represents a graded potential. So that graded potential occurs, and if it's enough, if it reaches threshold, negative 55, that depolarizing graded potential is going to trigger an action potential. And the action potential is going to propagate along the axon using voltage-gated sodium channels and voltage-gated potassium channels. That action potential travels along this long axon. This neuron enters the spinal cord, which we'll talk more about in the next chapter. But notice it's entering in the spinal cord. It's ascending up the spinal cord and it reaches a synapse. This is a synapse or a junction between the sensory neuron and a neuron known as an interneuron. Interneurons are the most abundant neurons. They're all in the CNS. The word interneuron means in between neurons because it often helps decide where these nerve transmissions need to go. So this interneuron is basically saying, okay, I need to send that into this area of the brain, which is known as the thalamus, which we'll talk more about when we get to the brain. And the thalamus is going to use another interneuron to relay that information to an area in the cerebral cortex, which is this outer gray matter of the brain. But we're assuming that this area of the brain is involved in interpretation of this stimulus on the fingertips. And there's processing of that information, probably using memory, because you have to remember what a pen feels like. There's probably some visual information going on as well. And then the decision is made to move that pen. That originates here in the cerebral cortex, but what we would call the motor area of the cerebral cortex. That motor neuron is going to transmit action potentials back down the spinal cord so the motor neuron can come out and stimulate the muscles at the neuromuscular junction. So notice we've got graded potentials occurring wherever the neuron is getting stimulated. Here it's getting stimulated by a neurotransmitter. Here it's getting stimulated by a neurotransmitter. We have black arrows representing the action potentials, which are transmitting, they're conducting or propagating down the axons. And then we even have a yellow arrow, which represents the muscle action potential. Remember that that has to occur in order to trigger muscle contraction. So that's the big picture. We're also now going to move into some other things that have to do with neuron physiology. One includes the function of myelin. We've talked about the cells that produce myelin. Oligodendrocytes produce it in the CNS. Schwann cells produce it in the PNS. But this gives us an idea of why and how myelinating an axon is going to speed up nerve transmission. In panel A, we have what's called continuous conduction. That's done using unmyelinated axons. So this axon doesn't have any myelin. We see that there's sodium going in, so this represents the depolarization phase moving down the axon. Over here in panel B, we have what's called saltatory conduction. Saltatory actually refers to the hopping of the action potential. The action potential, the depolarization, is actually prevented wherever there's myelin, but it occurs at the nodes of Ranvier. The nodes of Ranvier are the little gaps in between the myelinated regions that allows depolarization. So in essence, the depolarization hops from node to node until it reaches the end bulbs. And if you hop over large areas of the axon, you can get down that axon much faster. It's kind of like if you took baby steps, small little steps across the room, and compared that to making large jumps or skipping across the room. So you'll notice some numbers here. After 10 milliseconds, the action potential on the unmyelinated axon is about, I'd say a quarter of the way down the axon, whereas if you're myelinated, you're almost to the end. Okay, so some suggest that myelination will speed up action potentials by about 30-fold, 30 times faster if you have myelin. There's a condition called MS, a multiple sclerosis, where the immune system actually destroys the myelin, and that affects how fast nerve transmission occurs. So we should understand the difference between continuous and saltatory conduction and why it's so important for the nervous system. Next thing, I'd like to continue with this idea of velocity. So velocities vary quite a bit. Some neurons don't have to be fast. They do more maybe of the processing of information in the brain. They're largely gray matter, unmyelinated neurons. And then there are some that have to be fast in order to produce very quick responses. So we're going to look at some of the characteristics that can speed up nerve transmission. One is just the size, the diameter of the axon. Large diameter axons have less resistance, so less opposition to that flow of ions, and therefore they can be much faster. The next one we already talked about, myelination. Myelin is going to, again, prevent depolarization where the myelin is and allow the depolarization to occur at the nodes. It also prevents leakage of the charge. And again, we call that saltatory conduction, about 30 times faster than continuous conduction. So based on these features, these characteristics, there are three categories or classifications of nerve fibers. So again, there's a number of ways to classify neurons. Structurally, based on how many processes are extending from the cell body. Functionally, whether they're a sensory neuron, whether they are an interneuron or a motor neuron. And then we can also classify it based on their velocity. The three categories include group A fibers, which have large diameter axons and a lot of myelin, thick myelin. They're going to be the fastest. Some of our pain neurons are group A. So when you cut yourself, that very fast, quick perception of that intense pain is done using group A fibers. We also have myelinated motor fibers. So the somatic sensory neurons would be an example of like a pain neuron. And then our somatic motor neurons are also group A in that you can respond very quickly to possibly that pain stimulus. Group B fibers are kind of the intermediate velocity fibers. Their size isn't as big, intermediate diameter, and less myelin, and therefore a medium conduction speed. And these are often found in the autonomic nervous system. The autonomic nervous system typically doesn't have to respond as quickly as, say, the somatic nervous system. And then you have the slow fibers, the group C fibers, unmyelinated fibers, smaller diameter. We actually do have some pain neurons that are group C. That's the more dull pain that you might get, not the sharp pain, say if you cut yourself, but maybe kind of a nagging pain that might occur, kind of a chronic pain that some people experience, where it's not an intense, sharp shooting pain, but it's just kind of that dull pain. That dull pain is because we have group C fibers that are a little slower to stimulate. This picture just shows where our white matter and gray matter is. I'm going to go very quickly, but you can spend a little more time looking at where you would find a lot of the white matter in the spinal cord, which is around the outer edges. Gray matter is more internal. It's actually the opposite in the brain. Gray matter is most often found in that cerebral cortex. There are some internal gray areas called basal nuclei, but most of the white matter is found on the inside. Next lecture, we'll talk a little bit about some molecules out there that affect neuron transmission, and then we're going to actually get into the various neurotransmitters that are found.